Folio 120A is in Apple. If you don't get it, don't worry about it. I will give it back to you on the following Thursday. I will not have or make appointments for office hours next week. Right? That's my week to get stuff done. So basically Friday when I'm in there between 9 and 11, if there aren't way too many people, I'll be happy to answer questions as well if you have a few lingering questions about the final. The final will be cumulative, but again, look at big ideas and look at integration of ideas instead of the little details. How little and how big, I really can't tell you. Just focus on the big ideas. The exam will be fairly evenly distributed among all of the sections. So, about 20% on the slides for the first set of exam, 20% for the second, and so on and so forth. So, expect to have quite a few questions from the last set of exam as well. So don't think that just because you just took that you're not going to see that again. You will. Okay. That's it. And coming down, perfect timing. All right. So we are wrapping up. The last two slides are wrapping up information about the cell cycle. And even though I talked about the cell cycle going from quiescent, going through gap 1, gap 2, and then M phase, we said that some cells actually don't go through the gap phases, either 1 and or gap 2. Okay, and then by doing that, then the rate of cell proliferation will be much higher. Okay, so that's one thing. The other is also that some cells do not go through the gap. Some cells do not even undergo cytokinesis immediately. Eventually they will, because you don't have a lot of multinucleated cells. So eventually these cells will, but later on. But again, during the mitotic cell division, cells don't necessarily have to go through cytokinesis in order to complete cell division. Okay. All right. So we said that during gap 2, this is where materials are divided asymmetrically or symmetrically to determine whether the ultimate type of cell division is symmetric or asymmetric. But at the genetics level, how is that regulated? That's regulated mainly by methylation, by epigenetic alterations. Okay, so basically methylation of not just the promoter, but of DNA in general. But do remember that if the promoter is methylated, then that gene is inactive, right? So if we're talking about cells, especially cells that are differentiating, if they were to, if the differentiated cell is going to have fewer genes, active genes, then they are turned off by de novo methylation on promoter. If some of these genes that need to continue to be inactive from a stem cell to a daughter cell, then it's the perpetuating methylation, methylation by the perpetuating methyltransferase that's required. Okay? I'll just skip this. There's a neat story behind this, but we don't have time. This is another one of those slides, that don't get mesmerized by this. What this tells you is this, and don't even look at this because this is what it's telling you. So basically, the more potent a stem cell is, the fewer of the genes are methylated on the promoter. In fact, you would think then that the absolute most potent cell, which is a fertilized egg, wouldn't have any methylation on the regions at all, right? So then you would have all these genes that are active, ready to be used for any purpose. And then the more differentiated the cell, the more genes are going to be turned off. They are turned off by methylation on promoter regions. However, during differentiation, you might have genes that are turned on and turned off as well. So just by the total number of genes that are inactive or more when a cell is differentiated doesn't mean that once a cell is turned off in a stem cell, they can't get turned on later on. Does that make sense? So then now you have a different pattern of gene expression. So there are two different ideas there. The first idea is that the more potent the cell is, the fewer of the promoters are methylated. That's one. However, when a stem cell divides and undergo differentiation, right, even though the differentiated cells would have more inactive genes, but it doesn't. But an inactive gene in the stem cell it might get activated again in the differentiated cell, right? So just because a hematopoietic stem cell has gene A and B inactive doesn't mean that the daughter cell, the B cell, would have also inactive gene A and B. It could have inactive gene A, but gene B is active. But then the B cell would have more total number of genes that are inactive. That makes sense? So basically that's what this slide says. So that's the end of cell cycle and cell division. Are there any questions about the cell cycle, mitotic cell division, stem cells in general? Okay. We're going to wrap up the entire course by talking about wound healing. This is going to include genetics, cell molecular biology, anatomy, histology, physiology, and just any other ology that we can think of. Okay? Microbiology is going to subsume everything in explaining this. So the very first thing that we're going to do is we'll talk about a wound in the skin, at the skin level. So what is so that's the anatomical part. The histology is the fact that you have, we talked about the epithelium, right? This right here represents the epithelium, which is also called the epidermis. And you have these basal cells down here. Those are the stem cells that divide and differentiate into these cuboidal cells that eventually would get keratinized to form these keratinized cells, right? So that's what some people call the dead skin layer right there, okay? So that's the epithelium. This is the dermis, and the dermis is loose connective tissue that also contains sweat glands, sebaceous gland, hair follicle, lots and lots of blood vessels to provide nutrients and oxygen to the skin, and also nerve vesicles and muscles as well. Okay, and then below that is the hypodermis. Hypodermis is also loose connective tissue, and I think I said it the opposite yesterday. But this loose connective tissue is looser than the dermis, and it's looser because there's a lot of adipose tissue in there, so fat. That has a lot of fat cells in there. We're not going to be concerned with that. We'll just be concerned with the epidermis and dermis. Okay. That right there, although it's drawn and that looks pretty thick, but that's a very thin layer. So whenever you have a cut, right, a wound, it's not just going to wound up here. If you're going to have a wound up here, that rarely, if ever, happens. Usually when you have a wound, it goes all the way down here, even sometimes deeper, right? So when you have a wound like this, the very first thing to notice is that blood vessels are usually damaged. Okay, that's why when you have a wound, what happens? You bleed. Okay, so that also means that your epithelium is going to be open, and usually what's the function of the skin is to act as a physical barrier, right, to prevent stuff from going in. So now it's open, that means that stuff would come in, foreign materials, a lot of this room is nasty, right? If you were a microbiologist, you would know that it contains a lot of bacteria, microbes of all kinds, maybe even some pathogens that we don't even know of. So you have lots and lots of microbes going in. So the chance of you having a wound and not having microbes going in is next to none, right? So you basically, you can just think about the fact that anytime you have a wound, you will have microbes going in. So a part of wound healing is to activate your immune system, okay? So those two go hand in hand. So again, when you have a wound, it cuts all the way across here, okay? And it hurts because you also cut some of the nerve fibers there. So wound healing, there are 4 stages to wound healing, 4 overlapping stages. Hemostasis, inflammation, which is the immune system, right? The innate immune system. Proliferation, so that you can fill in the void, the spaces of the wound. And maturation, okay? Then we'll talk about scars. So roughly, this is what happens. You have a wound and you bleed, and then you have to stop the bleeding. Okay. Also, you have this open wound, so you have to block that. So that's the hemostasis. And then inflammation is the recruitment of additional materials, cells and proteins that can do 2 things. 1 is to heal the wound. 2 is to get rid of microbes. Right? So that's the inflammation or inflammatory response, period. Proliferation is to, when you have a wound, you're going to have damaged tissues, you're going to have an opening, so you have to fill it in by making more cells, right? So that's a proliferation part, to fill in the gaps, the void. And then maturation is to try to get the wound and damaged tissue as close to the original status as possible. And we're going to talk about the fact that whenever you have a wound, you will never ever go through wound healing and get back up to 100% of the function of that tissue. It's only a certain percentage, but not 100%. Okay? So we'll talk about that. So those are the 4 stages. They overlap. Okay? And so let's start by talking about hemostasis. So you have a wound. A little stab here is not enough. Let's say that you have a gash. Okay? You have a gash, and then you have damaged blood vessels. So we have to talk about something called resident cells. What are resident cells in the blood? Blood cells, right? All kinds of blood cells. In the dermis, or the connective tissues, there are also resident cells, right? Now, it's important to talk about resident cells is because we need to understand where they come from, where they proliferate. Just by a cell being in one place doesn't mean that that's where you can tell them to undergo cell division. Cell division might occur somewhere else, and that's exactly what we're going to talk about. So in the blood, resident cells are blood cells. In the dermis, the resident cells are the following: fibroblasts. Obviously, because fibroblasts not only are the resident cells, but they are the parenchymal cells, right, of loose connective tissue. They are the cells that produce the extracellular matrix, right? But then within there, you also have macrophages and mast cells, both of which are immune cells. And then you have some smooth muscle cells as well. Okay, so those are the resident cells. They kind of hang around, they anchor onto the collagen fiber through integrin. So I'm bringing back stuff that you already learned. Okay, so during hemostasis, this is what happens. Usually you have blood cells in the bloodstream and not in the connective tissue. One of those is platelets. Right? So you will only have platelets when you have a wound. But platelets are great because platelets then will induce a blood clot. Okay? So platelets, when they're in the bloodstream, they will not induce a blood clot. But when you have damaged blood vessels, the blood flows into the area of the injured area of the wound, and then platelets now normally aren't activated, right? So now when they're in the connective tissue, they get activated. How do you think they get activated? Anchorage onto collagen through integrin. And remember, communication can be done through cell adhesion, right? So again, bringing back stuff that we learned. So platelets are activated to release a number of different molecules, including a bunch of what we call clotting factors, factors that will produce a clot. And this particular clot is formed from fibrin, which is a type of protein, right? So we will call this a fibrin clot, fibrin blood clot. And this fibrin blood clot, again, it's activated or induced by the platelets that were activated by being in the connective tissue, right? The fibrin blood clot does two things. One is to, again, prevent further leakage of the blood vessel, blood from the blood vessel. The other one is to temporarily, very temporarily, block that opening right there, so that to prevent any more nasty stuff from coming into your wound. What happens if you don't have that blood clot there, or something's wrong with your clotting factors? Then you will continue to get nasty stuff in the open wound, and then you have nasty infection, right? Okay, and can lead to death if you don't take care of it. Okay, so that's what happens, is again releasing clotting factors, but also the platelets will also release a number of different signaling molecules. Now this release is just not a little bit of release, it's a huge dumping of all of these things, right? It's huge. So we call this type of secretion, that's a fibrin clot right there, with fibrin, with red blood cells, and then platelets are found in the area that will continue to produce these clotting factors to solidify that clot. There's a process called degranulation. Actually, let me go through the next slide. Degranulation. Okay. Degranulation is nothing more than the process of regulated secretory pathway. Okay, so it's regulated secretory pathway. But degranulation is a special name, is because you're not just secreting a little bit of material, you're secreting a whole bunch, right? So, for instance, a neuron will undergo will be activated to activate the regulated secretory pathway to release neurotransmitter, but you only release a little bit of neurotransmitter. That's not called degranulation. This is a lot of material being released, so it's called degranulation. Okay, and degranulation. Not all cells undergo degranulation. Platelets do. Okay, and again, it's a type of regulated secretory pathway. And when platelets undergo degranulation, again, some of the materials are clotting factors. Others are these growth factors right here. We'll talk about several of them. We won't talk about all of them, but we'll talk about several of them. Okay. We'll start by talking about these first two. So again, we're at the hemostasis stage, right? And during hemostasis, blood clotting is important. But also the platelets that are in the dermis right now is going to prepare or activate the next stage, which is inflammation, or the inflammatory response. And what it does is it secretes TGF-beta and PDGF. TGF-beta is a type of growth factor called a cytokine, and PDGF is a type of growth factor called chemokine or chemoattractant. So what happens is the cytokine TGF-beta is going to diffuse out and activate very specific cells, right, to recruit those cells to this site of injury. But they only activate. How do these cells know that this is the site of injury? Because you have the chemoattractant that attract these cells, right? So one ligand activates the cell, the other ligand acts as a beacon so that to call the cells over. Okay, so they work in combination. And what are these cells that we're talking about? We're talking about cells in the bloodstream. So again, now that you have a clot, so you no longer have leakage of blood, right? So then cells, not only in the bloodstream but elsewhere in the dermis, that cells have to be recruited to the area, right, to do work. So far, so good? Okay. So let's talk about actually resident cells first. The resident cells of the dermis first. Yes, sir.
If you don't have a clot, is it because you're lacking the fibrin or you're lacking the anchorage of the fibrin?
There are a number of different reasons. So hemophiliacs, for instance, there are a number of ways, different genes that are mutated. So it could be lacking the protease that form the fibrin clot, because fibrin is formed from a protein called fibrinogen that is cleaved. So you might not have that protease. Or some of the clotting factors along the way, there are 10 of them. Some of them are mutated. So if you don't have another pathway that can continue with that, then you have a problem. Could be, and there could be a problem not with the blood clotting, but with wound healing in general that can cause that as well. So there are a number of different reasons.
Okay.
So let's talk about resident cells. What are cells that need to be recruited to this area right now? One is a macrophage. What do macrophages do? Macrophages, they undergo phagocytosis. They're called phagocytes. And what do they phagocytose? They phagocytose. They're the cleanup crew. Right? When you have a wound, you have damaged cells, you have lots of stuff that come in. So they clean up the area, right? By undergoing phagocytosis. Again, cell debris and other material. In trying to clean up the area, they will also encounter, or they more than likely will encounter microbes. They also phagocytose them, but then that's an indication that the innate immune system has to be activated, because now not only do you have a wound, but you have an infection, basically, right? So now you have to clear the infection or clear the microbes, meaning you have to get rid of them. So macrophages act as a signal that, hey, more immune cells or the immune system, the innate, not the adaptive, but the innate immune system has to be activated. The particular type of innate immune system just happens to be inflammation. Okay, so macrophages is important in activating inflammation. But macrophages don't usually work alone. So again, macrophages will clean up the area and at the same time will help in activating the inflammation. Another resident cells that are going to be recruited is a mast cell, right? Mast cells, you probably have heard of them in reference to an allergic reaction normally, but they don't only work during an allergic reaction, they work during in activation and inflammation as well. Okay, so mast cells together with macrophages. Let me see if well, okay. Mast cells also undergo degranulation and release also signals that then will activate inflammation or the inflammatory reaction. Okay? And actually, and these are the possible things that mast cells, don't try to memorize this, I'm not going to go through all of this, but basically mast cells also undergo degranulation and release a boatload of stuff. Okay? Again, together with macrophages, they will activate the inflammatory response or inflammation. So first of all, what is the main function of the inflammatory response or inflammation? And the other one is, how does that work? Okay? So inflammation is the function of inflammation is to recruit more blood cells to fight against an infection. Again, indirectly, so it doesn't have much to do with, particularly directly with wound healing, but in fighting the infection, then you are helping the wound heal faster. Okay? So again, recruitment of blood cells to fight the infection, as well as recruiting additional proteins or plasma protein. Plasma is, think of it as the buffer within the blood, right? So recruit plasma proteins to, again, do the same thing to fight against infection. So that's the function of inflammation. And when you hear terms like edema, where you have leakage and then the area puffs up, right? That's the result of inflammation. Or you have inflammation, inflamed. Usually you think of it as that particular joint or area is inflamed or puffs up. It's due to the leakage of plasma material into the area. Okay? So that's inflammation. And again, it's activated by by macrophages and mast cells working in concert. So now it doesn't have much to do with platelets. Okay. So how does inflammation occur? The following steps occur. First, one of the very first event of inflammation is the blood vessels in the local area is going to increase in diameter. We call that vasodilation. Okay, so the blood vessels increase in diameter. When the blood vessels increase in diameter, what might be the result? The result is that you have a decrease in blood flow rate, which equates to a decrease in blood pressure in that area, but an increase in blood flow volume. So by having increased diameter, how fast blood flows is going to be decreased, but the amount of blood in the area is going to increase. And that's good because if you want leakage, you want to increase the volume, right? So again, inflammation starts with vasodilation, and that has two functions. One is to slow down the blood flow rates, but the other one is to increase the amount of blood volume that is there. Okay? So that the next thing that happens is that the blood vessels will be leaky. Don't worry about how they get leaky, just know that they get leaky. That means that materials now plasma and plasma protein can actually leak out, but also cells that have to come out can crawl out by cell migration through assembly and disassembly of microfilament. Okay? And there will be a slide later on that talks about rolling adhesion. We're bringing that back. So blood cells would undergo rolling adhesion and then migrate out, okay, by diapedesis. You've heard of these terms before, okay? And when cells that leave the bloodstream is called extravasation, okay? So again, inflammation does all of that to do what again? To recruit blood cells and to recruit additional plasma proteins to fight against the infection itself, but then at the same time a byproduct is to also help with wound healing. So macrophage activation. Okay. So what are some of these blood cells that are being recruited? Macrophages, more macrophages. Again, macrophages that are resident cells of the dermis would be recruited originally to clean out the area. Once microbes are detected, then more macrophages will be recruited to clear the microbe. So the function is slightly different, right? But macrophages that are recruited to get rid of microbes, are they going to clean out the area as well, or are they just going to say, well, I'm here to clean out the microbes, so I'm not going to clean up the cell debris? No, they're going to do all of the above, right? So having additional macrophages will help in both clearing the microbes as well as cleaning out the wounded area. Okay? Think about macrophages in the bloodstream. They're called monocytes. When they undergo rolling adhesion and then crawl out through the leaky blood vessel, and they're in the dermis, they're called macrophages. So that's just it. We don't even consider them to have differentiated because we're not talking about cell division, right? Cell differentiation and cell division go hand in hand. In this case, they just mature into, we just say that they mature into macrophages. So, reminder of rolling adhesion, remember there are E-selectin and ICAM here. E-selectin binds to carbohydrates on the surface of the cell. ICAM binds to integrin, remember? So now you have the adhesion, and then the cell squeeze through the pore, the leaky blood vessel. Okay, so when they leave, it's called extravasation, but the process, the mechanism is diapedesis. And let me just stop there. I'm also teaching immunology at the same time. I feel like I've said this 1,000 times, but have I talked about extravasation and diapedesis in this class? No. Okay, so when the cell leaves the bloodstream, it's called extravasation. It's spelled EXTRAVASATION. So extravasation. And then they mechanically, it's through diapedesis. That's the cell migration by crawling, right? Out of out. And it's spelled DIAPEDESIS. So the analogy is I leave the room by walking. I leave the room, that's extravasation. By walking, that's diapedesis, right? Pedesis, that's foot. Dia is tooth, right? So diapedesis. Then they're in the injured area, and then they walk some more toward whatever it is, right? Toward the cell debris or whatever it is in the dermis. Okay, so again, everything that we talked about in terms of cell migration by crawling applies here. Okay, so again, inflammation is vasodilation, leakiness of the blood vessel, okay, and then blood cells would undergo extravasation by diapedesis, and plasma proteins and plasma will just leak out into the area. Okay, to again fight. Very important to really inflammation. Its major function, its function is to fight against micro infection. The byproduct then is also to help wound healing, but the major function is only to fight against an infection. Okay. Other cell types that are recruited could be neutrophils. Neutrophils are also there to fight against infection, and that's all that neutrophils do. So macrophages, neutrophils. Okay. So that's inflammation. So again, hemostasis, inflammation. Now proliferation, the third stage. What is the function of proliferation? To reconnect or reestablish basically the connective tissue, which is the dermis. Because when you have a wound, right, you have damaged material. Now that material has to be reconstructed or reestablished. You have to reestablish the epidermal layer or the epithelial layer. The same thing, epidermal or epithelial, same difference. Okay. And then reestablishment of the blood flow, so make more blood vessels. So all of this has to do with proliferation, cell proliferation. To reestablish a connective tissue, you have to make more fibroblasts, right? So fibroblasts have to be told to undergo cell proliferation, and on top of that, the fibroblasts also have to be told to secrete more extracellular matrix. So that's the reestablishment of the connective tissue. The reestablishment of the epidermal layer, then the basal cells have to be told to undergo cell proliferation and then differentiation to reform the epithelium. Yes, ma'am.
Sorry, could you say that whole last part again?
Yes. So to reestablish the epithelium, then basal cells have to be told to undergo cell proliferation and differentiation, right? To reestablish the epithelium. So again, we're talking about asymmetric cell division here. And then the blood vessels have to be told to the area has to establish or form more blood vessels, right? So let me come back here. So fibroblasts that are told to undergo cell proliferation to make more fibroblasts to reestablish the connective tissue will be told to undergo cell proliferation by three different things, by FGF, but also by both TGF-beta and PDGF. So TGF-beta and PDGF have double duties, right? So all three of these are involved in telling the fibroblasts to make more fibroblasts by undergoing cell proliferation. the reestablishment of the epithelium, the epithelial cells are also told by TGF beta and PDGF to divide, right, and differentiate to reestablish the epithelium. And then more blood vessels are the area your body is going to be told to make more blood vessels through VEGF, which is vascular endothelial growth factor. Okay? So now this will bring back the idea of signaling, right? Ligand binding to receptor should bring back the idea of signaling systems, which is most of this would be the paracrine system, and also signal transduction pathway, and then leading into cell cycle regulation by inducing the expression of MYC, right? And then the cell cycle, and then the regulation of, and then cell division. All of that is plugged into here. Okay? So that's proliferation. So problem. When you have, at the beginning, you have a blood clot, right? Oh, when you have a wound, yeah, you have a blood clot. And the blood clot, its function is to, again, plug up the blood vessels so that you don't have any more leakage. If you have a problem with blood clotting, you can bleed to death when you have a wound, right? So that's not good. You also plug up the surface to prevent any additional material from flowing to the injured area. However, a blood clot is only a very extremely temporary barrier. It would be produced almost immediately within seconds or minutes, and it will only last for a few hours. Right? But you need a more permanent, not completely permanent, but a more permanent barrier, and that is a scar. A scar you see, right, is a more permanent barrier. Okay, and that scar is formed by mainly, let's see, that's the next one. Okay, formed by it contains mainly collagen, so it's called a collagen scar. And the collagen scar is secreted and produced by fibroblasts. Okay, so that's why you need more fibroblasts for reconstruction, right? So for proliferation, not only to reconstruct the dermis, but also to form this scar at the wounded area, right? So it's more permanent. Some scar lasts a few days, a few weeks, others last a few months or years, and others last a few decades. It depends. Right? It depends on the wound, the severity of the wound. It depends on the effectiveness of the wound healing process. And it also depends on external, what else is going on. Do you have an infection at the same time? Do you have some kind of a genetic, hereditary problem with wound healing? And we'll talk about that. Okay? So, but fibroblasts are important in producing not only to reconstruct the dermis, but also to produce the scar tissue. Okay? So again, fibroblasts is induced to do this by FGF and TGF beta and PDGF. Just a reminder about migration. Anytime that we talk about migration, it's by crawling, and so that should bring back all of the activities about microfilament assembly and disassembly, and all of that stuff. That again, I always hope that none of this gives you nightmare, but you need to remember. You need to remember everything about the communication through integrin. Okay. Just a reminder, rack and roll. Just a reminder, when a cell is told to undergo cell proliferation, there's hydrogen that leads to the expression of MYC, and then that leads to everything else. So those are just reminders. And then here's a scar matrix. It's composed of this, but again, mainly collagen, so that's why it's called a collagen scar. Okay? Whereas a blood clot contains mainly fibrin, so that's why it's called a fibrin clot.
GAGs, proteoglycans.
Okay. So again, reestablishment. So a scar would be here, right? Reestablishment would be cell division of the basal cells here and also differentiation. And these cells will not only move upward as normal, but also fill in the space in here. So they have to migrate laterally as well. So again, through migration by crawling. Okay? So these cells will do that. And the scar is great because a part of the scar will also be used as the substratum for these cells to walk. Okay? Okay. So if you have a big old scar, right here, right? Let me see. There are a couple of different. Oh, okay, yeah. So eventually you can reestablish the wound area. Usually the scar, most of the scar, if not all of the scar, will go away, right? But that's a long drawn out process. So this is now, we're leading into maturation. Okay? So proliferation, maturation. And proliferation and maturation, even though they overlap, but they may continue for a while because as the scar, how does a scar go away? Macrophages. Macrophages will continue to be in the area, chipping away at the scar and also undergo phagocytosis, right, to get rid of the scar material, which is mainly collagen. So macrophages will hang around, chip away, and then the area then would have to be refilled with the tissue, mainly the dermis. So then fibroblasts also have to hang around to secrete the material. And again, the maturation process is again this chipping away at the scar and filling into the void, right? Mainly done through macrophages and fibroblasts. Okay? And this actually is an indication of, this is the dermis here, this is the hypodermis because these yellow things represent adipose tissue. Okay? We good so far? And again, all at the same time, the only thing that I want you to know about this area is that blood vessels have to be formed. Right? Additional blood vessels have to be formed to replace the damaged blood vessels, and that is directed or activated by VEGF. That's it. Done. Right? Don't need to know any other details about this. Just VEGF activates and induce vasal. The other term is the term angiogenesis. So the formation of new blood vessels is called angiogenesis. ANGIO, and then genesis, GENEO, ANGIO, GENESIS. So angiogenesis is the process by which new blood vessels are formed. There are different types of angiogenesis, and we won't get into those, but they are directed by VEGF. So again, I'm making connection between signaling and effective function here. An example of normal, this is before injury, and then if you have an injury, and then angiogenesis occurs, and you make new blood vessels. That's what it looks like. Okay. So we talked about proliferation leading into maturation. If you look at the entire process, all four processes, you will see that you have a wound here, and hemostasis occurs almost immediately. Right? Within, again, like I said, seconds, really minutes after you have a wound. And then that probably would be it, right? And then inflammation takes over because can you have a wound in the wound healing process without inflammation? Probably, but not likely at all. At all, because again, practically all the time, right? 9.999999999% of the time, you will have microbes entering the wound. So therefore, you will practically always have inflammation. So most people who study wound healing just pretend that it always happens because they have never seen a case where you have a wound and not have inflammation occurring. Okay? But again, the function of inflammation is not directly due for wound healing. I want you to understand that, even though it always happens, but it's not directly there for wound healing. It's directly there to clear microbes. Very important thing to remember, right? So inflammation occurs, and it takes about hours to maybe a day or so for inflammation to last. And then inflammation then has to do with recruitment of neutrophils and macrophages in general. So that's why you see a spike here. And then eventually neutrophils are no longer needed if the microbes are cleared. So again, inflammation is a part of the innate immune response. If for some reason the microbes continue to be there, then that's where the lymphocyte line comes in. So that's a part of the adaptive immunity. So then it peaks roughly after inflammation occurs, and then it will last as long as necessary for the adaptive immune response to get rid of the microbes. And usually that takes about an additional 2 or 3 or up until about 4 days or so. Yes, ma'am?
Okay, what are the two immune responses you're talking about?
So innate immune response is inflammation. Inflammation is a type of innate immune response, and that basically always happens during wound healing. But adaptive immune response may or may not occur depending on how quickly your body or how efficiently your body can get rid of the microbes, which is independent of wound healing, right? But it's still here just to show you that there are two different major types of immune system, okay? Back to wound healing, macrophages are going to be present to, first of all, get rid of the microbes, but also to clean out the area and remain there throughout maturation to do what? To chip at that scar, right? So that's why you see that it doesn't, here's macrophage, it doesn't quite go away. It's going to be at the basal level for two reasons. One is to chip at the scar and to continue with maturation. The other is that some macrophages are now resident of the dermis as well. So the amount of macrophages never dips all the way down to 0. Okay? Fibroblasts will continue to be there again because during maturation, then as the scar is being chipped away, removed, the dermis has to be filled in. Okay? And that's done by the fibroblasts. So that's maturation. And that maturation can occur, might take a few months, might take a few years, might take a few decades. Okay, however long. And again, it has to do with the severity of the injury, the effectiveness of the wound healing process at the time, as well as if you have other problems going on. And so now we're going to talk about the other problems that go on during wound healing. Hopefully that's the next slide. Oh, not quite. Almost. Almost. Talking about all 4 stages, right? So within that, during wound healing, especially during proliferation and maturation, but during wound healing, you have, let's say, talk about your skin. You have an open wound, right? Your skin is very taut. It's stretched out. So if you have a cut, what does that do? That releases a tension, and if you don't do anything, what happens to the skin? It tries to pull out more and more and more and more, right? So during all stages of wound healing, the edge of the wound will have to be pulled in. Because if it's not pulled in, again, it gets bigger and bigger and bigger and bigger because the skin will try to pull it out, right? Because it is taut. Okay? So your body will have these cells called, now this goes, your body would have cells called myofibroblasts. They are fibroblasts, but they have a function very similar to a muscle cell. myo always refers to muscle, right? So a myofibroblast is a special types of fibroblast. So a cell has to be told to take a fibroblast, divide and differentiate into a myofibroblast. Okay? So fibroblasts themselves may be adult stem cells, but they have extremely low potency. So these myofibroblasts, what they do is they grab a hold of the edge and on all sides and contract so that they pull the edges in, while fibroblasts will produce a scar, right? While proliferation occurs so that additional the reestablishment of the epidermal layer, the dermis, connective tissue, as well as angiogenesis occur, right? So myofibroblasts have to pull them in. Okay. Now, if there's a problem with, and this is called wound contraction or wound contracture, is naturally done through myofibroblasts. Now, if you have a surgery or something, of course, surgery is the wound is going to be a lot bigger, and at the end of surgery, you do want your skin and your tissue to heal better, so you would have synthetic wound contraction mechanism by a staple or a suture, right? So that's why that is. So again, if you don't, then the skin would get stretched out more and more and more, right? Okay. Now, if your skin gets stretched out more and more and more, that's okay. Your body has a built-in mechanism, and what is that? Is scar. So if it's not pulling in, any void, any space that you have would be filled in by scar, and that's a problem. If you have a scar that's way too big, right? If you have a scar, I see a few cringing, so I'm going to move that out. I used to work in the hospital, and I have the worst thing that I was told not even see, because I'm glad I didn't see it, and I won't even tell you because you might throw up. There's this one case that it was so horrible that even I didn't want to see it.
Now you
But
gotta
I'm
say.
used to seeing those things.
You gotta tell us.
What is it?
What is it? I'll tell you at the end. If we will have a few minutes, and I'll let the people who are going to vomit leave the room, and I'll tell you the story. But and it will remind me. But what happens if your edges, the wound edges, don't pull in again? Your body fibroblasts will be told to divide more and more and more, right? And then produce more and more extracellular matrix to form a bigger and bigger scar. Well, you don't want that, because the bigger the scar, the more difficult it is to mature, for the wound to mature and heal, right? So then you have a problem. So you do want that contracture. So at the end of surgery and everything, you need staples and sutures. Yes, sir?
How do those myofibroblasts control calcium being open?
What do you mean control calcium?
So that they're contractile, right?
Right. Oh, what is the mechanism of that? Well, they perform, their function is very much so like smooth muscles. So they control that way. So the calcium source is what you're asking, is from both the extracellular matrix and the ER. But again, the contraction is not all or nothing, it's graded. So if the skin was being pulled outward harder, then the contraction is more. If the skin is being pulled out a little bit by just a small wound, then the contraction is less. And then, so that's a good question. So that's a connection right there. So that's why myofibroblasts look and function very similarly to smooth muscle cells. They just happen to have, the origin came from different places. Smooth muscles came from myoblasts. Myoblasts came from fibroblasts, actually. Okay, so that's a good question. A wound will never ever, that I know of, a tissue that has been wounded will never ever recover 100% of its function, especially if you were talking about skeletal muscles. Okay, so that's enough said. So again, a damaged tissue will never, that we know of, recover 100% of its use. So healing abnormality, again, you have a wound that will not heal properly. Okay, why? What are some of the reasons for that? There are two major reasons. One is the failure to heal. It just doesn't heal at all. And the other one is there's healing, but there's way too much scarring. Okay? So let's talk about one first. Let's talk about the failure to heal. Most of that is due to excessive inflammation. So if you remember, inflammation, the reason for inflammation is not directly for wound healing. Right? It's not. But it's always there. And now, how would you have excessive inflammation? Two of the major reasons why you might have excessive inflammation is one is by the presence of a biofilm. Wow, all the way to from September that we talk about biofilm. Remember biofilm has to do with bacteria, right? So these community of bacteria that secretes its own, their own extracellular matrix, they live there, they're happy. It's a whole community. There's no reason for them to be removed. It's because the immune system, the inflammation, there's something wrong with the inflammatory response. There's something wrong with your innate immune response. There's something wrong with your immune response in general. So that's why these bacteria just take up residence here. And if they take up residence here, the macrophages and the immune system can't get rid of all of this because, again, proliferation and maturation can only occur if this area is cleaned out. If it's not cleaned out, then wound healing will not be completed. Yes, sir.
I have no idea about the pharmacology of anti-inflammatories, but if we take anti-inflammatories, does that suppress our immune system and make us susceptible to this type of
Yes, if you take an anti-inflammatory, then that will reduce the activity of mast cells, right, as well as macrophages. And let's say antihistamine. You remove histamine and the cell can't dilate, and the blood vessels won't be leaky, so you're not recruiting more cells, right? You're not recruiting more blood cells as well as blood proteins. So yes, NSAIDs are not good for you to heal a wound. Right? Because why? Because of this. But on the other hand, if you're not able to get rid of that infection, you're not going to have wound healing to begin with. So which one do you want first? Chances are you want to heal the infection first, unless you have an infection somewhere else and you need NSAIDs. That makes sense, right? So again, it's depend on the location as well, right? So if you have an infection right here, the wound is not going to continue because it stops basically at the inflammation part because it has to clean out the area first before proliferation can even get started. Yes, ma'am.
Kind of along those same lines, so what causes inflammation when there's an injury where the skin did not break? Like when you sprain something, it swells, right? That's inflammation. So
Yeah.
what causes
So
that?
then internal, so if you have a sprain, you also have internal damage.
Okay.
So damages in general will cause that. And blood vessels, especially capillaries, are very sensitive. They're very fragile.
Mm-hmm.
However, there are multiple means to induce the inflammatory response as well. So it's not always from platelets. It can be due to other processes as well that we don't talk about. So that's an excellent question. So two things again. If it's internal, you can have internal bleeding. Or you can have other signals that are activated. And mast cells aren't always activated. So that's one. How about people who have chronic inflammation? And there's a whole list of stuff here. Don't try to memorize this. Don't try to even remember one of them. Just know that chronic inflammation, okay? But Crohn's disease, ulcerative colitis are two of the major ones. But cancer, some cancers are involved with chronic inflammation, arthritis, osteoporosis. So if you have chronic inflammation, these folks, I don't know if you know people who have some of these diseases or disorders, but they have a really hard time to heal a wound. Okay, I think two or three more slides. So the other is problem with excessive scarring. That's also a wound healing problem, right? Abnormal. There are two types. One is called hypertrophic scar, and the other one is keloid scar. And I was informed that on the, one of the exam, practice exam question that I chose the wrong answer, which I checked and I did choose the wrong answer. And so there are reasons why, but that doesn't matter, I just chose the wrong answer, okay? So the correct answer is hypertrophic scar is due to the failure of contraction by myofibroblasts. So it's not just fibroblasts, but it's myofibroblasts. So either if you don't have enough myofibroblasts, so if you have a condition, and people who have, you have a condition where myofibroblasts can't be produced in great amount or at all, or the myofibroblasts somehow can't contract well enough. Let's couple that with people who with osteoporosis. I'm just making up something, right? If you can't deliver enough calcium and sequester enough calcium in the extracellular matrix, then it's really hard for the myofibroblasts to contract, right? Then the wound, the edge of the wound would get bigger and bigger and bigger, and to fill in that hole, then you have a bigger and bigger scar. That's the idea. Okay? So that's hypertrophic scar. Versus a keloid scar, which is a problem not with the myofibroblast, but with the fibroblast. And fibroblasts, how do they receive information? By information to differentiate and secrete scar tissue through TGF-beta and PDGF and FGF, but more specifically TGF-beta, right? You have TGF-beta in the area. Now, a fibroblast may have a few TGF-beta receptor or a lot. If it has a few TGF-beta receptors, doesn't matter how much ligand, how many ligand molecules TGF-beta is in the extracellular matrix, the fibroblast is only going to be told to secrete that much scar tissue with a limited number of receptors. But if the fibroblast, there are conditions where people who might have a lot, you're talking about 2, 3, or 5 times the number of receptors, TGF receptors on the surface of fibroblasts, what happens? Then the fibroblast is going to be told to produce an excessive amount of scar tissue, right? With, again, the same amount of ligand, okay? And that then you would have a problem called keloid scarring. And the very last slide of the last day of lecture is that only people who have this problem, unless you can work around it, can actually have pretty tattoos like this. This person cannot possibly have this nice looking tattoo unless, I can't tell whether it's he or she, it looks like it might be a she, cannot have this nice pretty tattoo unless this person has a problem with excessive expression of TGF-beta receptor. Okay? And so people actually use this condition for art. Okay? So you want to hear the story? Or you want to
That's nasty.